Confused with each other? You haven't figured that out already? Epididymis and the testis, when you look at the seminiferous tubules, can be confusing, all right? So if I were to show you a slide like this, would you be able to tell whether this was epididymis or the seminiferous tubules of the testis? What do you think it is? And it's okay to be wrong. You're still studying. Anybody have a guess?
Epididymis.
Epididymis. All right, now let's look, because the epididymis is a single tube, and you're correct. The epididymis is a single tube, but it's highly coiled. So when we cut through it, we see multiple lumen that have been cut. Seminiferous tubules, multiple tubes that aren't as highly coiled, and so we're cutting through multiple tubes at once. So compare, those of you that weren't sure, compare this slide to this slide. It's not a very good slide at low power. Notice all of these purple circles. Those are the nuclei of the developing spermatozoa and spermatids and spermatogonia and so on, all right? They're moving, the spermatogonia are down here near the outer edge of the connective tissue around the seminiferous tubule, and then they move to the very surface because that's where the late spermatids are associated with the apical plasma membrane. If we look at the epididymis, it's a pseudostratified columnar epithelia. So we do have multiple layers of nuclei because that's incorporated in the pseudostratified, but this area of the cells is clear. There aren't any nuclei there. That's the apical columnar portion of those cells. Now you might say, well, when I looked at it in the microscope or as an image, I see sperm tails sticking out. Those aren't sperm tails. The flagella, those are long microvilli that we see in the epididymis and the Vas Deferens. Okay? So how do you know this isn't Vas Deferens, which also has pseudostratified columnar with long microvilli? Vas Deferens is a single tube, but it's not highly coiled. So it will be cut in cross section, but you will see just one lumen. Okay? So it's pseudostratified. Here you can see the longer microvilli, all right? You might even see some sperm in the lumen. It's too close to see, but you have a very thick muscular, several layers of smooth muscle around that. How do you know this isn't the spongy urethra? What system in the body is the spongy urethra a component of?
Urinary.
Digestive?
No?
Urinary. Urinary.
No, well, it's reproductive in male. It's not reproductive in female. What is it in female? Pardon?
Excretory.
Yes, but we have different kinds of excretory. Digestive is excretory, and respiratory is what passes through the urethra, either the male or the female.
Urinary.
Urine. So it's part of the urinary system. For males, it's described sometimes as genital urinary, so genital and urinary. Anybody remember what the characteristic epithelium is for the bladder, the urinary bladder?
Transitional.
This is back from like the third or fourth week in Bio 430s, but might have been a while.
Transitional.
Transitional, okay? So you're going to have those, kind of looks like raindrops, vertical layers of cells rather than a horizontal and stratified squamous. It's going to have layers of cells all the way to the surface, and they're going to be typically rounded cuboidal cells at the surface. A seminal vesicle. Okay, you don't have to know the histology for the bulbal urethroblast. Take a close-up look for the prostate columnar cells. They kind of look, well, they're pale. You can see the secretions in them. And this is a little close, but the openings of the seminal vesicle, this is a lower power view, but the cells are darker. And notice there's lots of openings, not with very little connective tissue between them like the prostate gland has, okay? And these cavitations are contained, they're closed. When we get to female next week, which won't be on the quiz, but will be on the lab exam, this is the oviduct. Looks very similar to the seminal vesicle. But in the oviduct, it's like a maze that you can always get out of. You're not trapped in those little ovals and so on, okay? So looking for those differences can help you go, oh, wait a minute, And I know it looks like this, but let me look for these specific characteristics. All right, so that can help you differentiate between things that can look similar. Look for the things that you know are characteristic of a particular one. If you don't find it, it's going to be the other organ that you're looking at. Okay? So I talk about that in that reproductive, male reproductive, that makes testosterone. What would we call it? A diploid cell or a haploid cell? Diploid. So for the human, how many chromosomes total are in a diploid cell?
23.
Do you know? Diploid cells are called 2N, where N equals 23. So we have 23 chromosomes that came from the sperm, and 23 chromosomes that came from the egg. And when the sperm fertilizes the egg, we get a cell with 46. Okay? So when we say N equals 23, we're talking about how many different types of chromosomes there are. All right? It's like trisomy of 21. There's a third copy instead of 2 copies of chromosome 21. So chromosome 23 is our X and our Y, or our X and our X if you're female. So I'm not going to draw 23 pairs of chromosomes. I'm going to just use the X and the Y. Okay? So in our spermatogonium, which in the male start mitosis again. Mitosis doesn't occur after birth, and it starts again at puberty, so the male continues to make more spermatogonia. In the female, it stops before the female is born. Okay, so there's no more, there shouldn't even be any more oogonia. The female is born with primary oocytes. So a spermatogonium is 2N. It's a diploid cell, and so it has a Y chromosome from the sperm that made this person whose testes we're looking at, and it has an X chromosome from the egg that original sperm fertilized, and that's what we'll be looking at. Okay, so in regular mitosis, where you have daughter cells that look exactly alike, when these chromosomes replicate and form sister chromatids, they would line up like this during metaphase, where they separate, okay? Anaphase, rather, where they separate. And the microtubules would shorten and would end up with the same chromosomes as we started with in our 2 spermatogonia. They look exactly alike as far as chromosomal material is concerned, still 2N. When our spermatogonium undergo mitosis, typically one remains behind to undergo mitosis again, and the other daughter cell, so one would remain behind, and the other daughter cell would become I don't know why they're called daughter cells, because they're son cells. The other one would start to become a primary spermatocyte, okay? So when that happens, we start meiosis, and the chromosomes replicate. But instead of staying as sister chromatids, they form, what do we call this? Tetrads, okay? So I want you to be aware that the tetrads exist in the primary spermatocyte. Because of that tetrad arrangement and the close proximity of the chromosomes, we have crossing over, which is the exchange of genetic material between the chromosomes from the egg, the mother, would be the grandmother of this new baby on the father's side, and the chromosomes from the sperm, from the guy that would be the grandfather of this new baby. And it's totally random, okay? So some may have a lot of crossing over and exchanging of genetic material, and some may not have very much. It's amazing how the daughter or the son of a couple can look almost like an identical twin to their parent. And other times, they don't look like them at all. Okay, that's from this random crossing over. Now, so that's called crossing over. And again, that occurs in the primary spermatocyte stage. Notice there's no cell division between spermatogonium becoming a primary spermatocyte. It's all internal chromosomal changes that occur. So if this was 2N, is this still a diploid cell?
No.
Yeah.
Yeah, because we still have the chromosome from the father and the chromosome from the mother. That's what the 2N means, okay? So it's still diploid. However, in my nomenclature and patterning number, I want you to recognize that we have 2 copies of all of those, okay? So it's still a diploid cell. And on your lecture exam, I'll have a little table. Your lecture exam will be about 65% multiple choice and about 65 to 70, and the rest short answers. Not an essay, but short answers. And one of those short answer types would be identify the chromosomal pattern of these named cells. And I'll give you the 2N, 2 parentheses in, 2 parentheses, 2N, and so on. If you just put 2N for a primary spermatocyte, leave that off, it won't be wrong, but it won't be as much information as I wish. Okay, so you would get half credit for that. Because it is a diploid cell, it is 2N, but it's got those extra copies, the double copies of each. All right, now we're ready for our first meiotic division. Anybody have questions on the division of meiotic cell division? In the egg, the same type of division occurs, but it's unequal in size. So while for the sperm, when we go from primary spermatocyte and divide to form secondary spermatocyte, we form equal sized cells. In the female, that second cell is really little, but the chromosomal pattern is still the same. So when we have this process in the male, we have 4 spermatozoa from 1 spermatogonium. In the female, we have 1 oogonium, ends up with 1 ovum. So we kind of waste the genetic material of those other cell divisions. But we'll look at that next week. All right, so this is a secondary spermatocyte. Remember, that's the one that we don't see in the microscope? So a secondary spermatocyte will never be a correct answer for an actual histology picture. They just don't last long enough to be captured. All right, so separation occurs now. Do we have both an X and a Y in each of these cells? We have only X's here and only Y's over here, okay? Now that separation is totally random. This is for chromosome 23. For chromosome 10, the pair of chromosomes that end up here may be from the mother. And the other chromosome 10 ending up here would be from the father. Okay, so how that gets distributed is totally randomized. But both of the pairs are still attached to each other. But we don't have both the male and female contributions. We only have one or the other. We might have the male for one and the female for the other one, and back and forth. We might have out of 23, we might have 15 from the father and what's 15? 8 from the mother, so it's totally random. But we have split that relationship, and so this is only N. We have 2 copies, so I prefer to write it like this to remind ourselves that we have 2 copies of each N, okay? And so because we only have an N, this is considered a haploid cell. So at the end of our first meiotic division, we now have haploid cells. We're not done yet, but the whole purpose of meiosis is to reduce the number of chromosomes. Because if we had a sperm with 46 chromosomes fertilizing an egg with 46 chromosomes, it would die, okay? And many other times, if there's even 4 or 5 chromosomes that didn't separate here, it doesn't develop. In trisomy of 21, the 21 chromosome for one or the other didn't split. So we have 1 chromosome 23 from one parent and 2 chromosomes 23 from the other. And that's how we get that misnumbering. All right, second spermatocyte. First meiotic division gives us our haploid in, but we still have that separate copy. So in the egg, this would be really, really tiny, but it would still have all those two parentheses in chromosomes. All right, one more meiotic division. This would be now a separation of each of those chromatids. And now we just have one Y. So this would be still a haploid cell. We have this one X. So still a haploid cell. Whoops, the right X. And we don't put any parentheses around it because we have just one copy of each of those. What comes after the second spermatocyte, after our second cell division? The early spermatid. Okay? So the early spermatid is our truly first haploid cell without any extra copies. So first meiotic division here. Second meiotic division here. Early spermatid. So no more cell divisions, all right? What we do now is it changes from that round cell that looks like this, and we start losing cytoplasm and losing organelles until we actually look like a spermatozoa. The late spermatid. It's called the late spermatid when it's still associated with the Sertoli cell. We don't call it a spermatozoa until it's actually out in the lumen, okay? But it's only losing cytoplasm, it's not losing any more chromosomes. So the spermatozoan, whether it's the late spermatid, the early spermatid, or truly mature spermatozoa, is still going to be a completely haploid cell.